the genetic material, which is DNA, or RNA in some cases where we use RNA viruses. We engineer RNA as well, or for RNA vaccines, etc. And the second is bioprocess engineering. So to do anything with the genetic material, we need an environment which has to be sterile. So that is bioprocess engineering, maintaining a sterile ambience in which you can do the chemical engineering process. And then we talked about a little bit of about DNA and plasmids. And plasmid is a very important thing here because if it was not for plasmids, we could not have evolved the field of biotechnology. So both of you know what is plasmid and why it is special and where it comes from, right? Any doubts about plasmid?
No.
Okay. And then another important thing was restriction enzymes. We talked about how Herbert Boyer um, figured out restriction enzymes from E. coli bacteria. You know what is a restriction enzymes, right? These are of two types, endonucleases and exonucleases. Mostly for biotechnology, we use endonucleases, and exonucleases is also used, but for purification processes, not majorly for making a recombinant DNA. So endonuclease is important, and it leaves sticky ends. All those things we discussed. And then cloning, DNA cloning. So making identical copies of any DNA template is called DNA cloning. And how plasmid helps us in that is because it is autonomously replicating. So the benefit is that it does not need, uh, uh, you know, it does not need to rely on the genetic material of the organism, genetic material of the bacteria, which is the nuclear DNA. It can independently of the nuclear DNA replicate. And we also studied that for replication to occur independently, any piece of DNA should have what? What facilitates replication of any piece of DNA? Remember, RO, there is something called origin of replication, ORI, right? So
Yes,
any
sir.
piece of DNA that needs to be replicated in a living system needs to have a sequence. So that sequence, just think of it like a gate pass. So to undergo this process, you need a gate pass, and that gate pass is origin of replication. If you don't have that origin of replication sequence, then the machinery won't recognize that piece of DNA as an important one that needs to be replicated, and it can't be replicated. So plasmid have their own origin of replication, and that's why this piece of DNA is interesting. See, otherwise, if it was just about making a circular DNA and sending inside the cell, um, it was not that difficult to achieve. But the problem is why plasmid is special is because it has all the components that make it self-reliant, autonomous, right? Okay, then we talked about restriction enzymes in detail. As I told you, exonuclease, endonuclease, they are called molecular scissors and how they function, etc. And then we came to tools, which is again, these restriction enzymes are tools. We talked about how they are named, etc., and their action, how they leave sticky end. I hope there's no doubt here, any doubt? On how restriction enzymes work and how they, you can just look at this diagram, like take some time, look at it and tell me if you understand it. If you see it and you understand it, then you probably know the concept. If you see and if you're confused, what is it? I don't recollect. Then maybe you forgotten. Okay, perfect. And cool. Then we talked about the the steps. So we take a foreign DNA and using the same restriction enzyme, cut both the foreign DNA and the vector DNA. And using ligase, we join the DNA after introducing our piece into it, and then we send it into the living cell, which is called the process of transformation. So first is restriction digestion, second is ligation, third is transformation. Then we culture it. Culturing is basically cloning, because as the bacterial cell divides, it also makes multiple copies of our plasmid. And once that has happened, then we purify, do a large scale production of that protein or that bio product, and then we sell it. In that, a very important aspect is when we do restriction digestion, we have to separate our DNA fragment, like the DNA of, what do you call it? The DNA of interest, the foreign DNA of interest that we are interested in. We have to separate it from other pieces of DNA that we don't need. Basically, our gene we need. And for that, the process of gel electrophoresis helps to separate DNA fragments. And on what principle it works, do you remember? Because this is a very important concept. So on what principle gel electrophoresis work?
So sorry.
On what principle gel electrophoresis works?
So that on electric on passing electricity, the shorter particles will move the farthest. Is it?
Yeah, but why? What is the principle behind it? So DNA, as a molecule, it is negatively charged, right?
Yes, sir.
Yeah. So when we apply a electromotive force, then what happens? Due to the charge, if there is a potential difference, which means there is a positive terminal and there is a negative terminal, then DNA being negatively charged will be repelled from a negatively charged terminal and will be attracted towards a positively charged terminal, right? This
Yes.
is first thing. So the force is given by electromotive gradient, but
Yeah.
on what basis the separation happens? Because when you say it that on the basis of electricity, the fragments will be separated, but electricity is just pushing all the fragments. So it's pushing all the fragments like the same. So on what basis are they separating in that gel? They are getting
So the
separated on the basis of their size, how big that fragment is, right?
yes.
Because bigger the size, greater will be the charge on that size, on that fragment, correct?
Yes.
Where does charge comes from on a DNA? What gives negative charge to a DNA? Remember we talked about structure of DNA in chapter 4, kind of. What gives so phosphate group PO4. Remember there was one phosphorus end and the other one was a carbon ribose sugar, right? So
Yes.
phosphate and sugar makes the backbone and bases protrude towards the inside, right? In a DNA. So
Yes.
the more number of phosphate groups, the higher will be the negative charge on DNA. So if a fragment is bigger, it has more negative charge, and the force exerted on that will be more. Plus the gel that we use, it's called gel electrophoresis. The gel has pores, pores of a particular size. And if there are fragments bigger than that size, they will find it difficult to move through the pores. So they will move slow, but smaller DNA fragments will move faster. So the smallest fragment will be the farthest from the well where we loaded it. And the biggest fragment will be the closest to the well where we started, right? So on this basis, we can separate DNA fragments. Make sense? We talked about how can we change the size of the pores by increasing the concentration of the agarose. So this gel is made up of agarose. Agarose is a biopolymer and we get it from seaweed. And when you heat it, it is liquid. And when it cools down, it solidifies and then it forms a cross network of fibers. And because of which there are pores in between left. Right, Hiram and Parker? Is it clear? Any doubts in gel electrophoresis? Okay, perfect. Parker, do you have any doubts? So these are techniques which obviously will be asked every year. One or the other question in board or entrance for sure is going to be on the concept. Okay, then we talked about cloning vectors like the plasmids, synthetic plasmids that already have all these sites for restriction enzymes to work. So they are called restriction sites. And this is where so this like comes pre-made, and based on your experiment, you can conveniently choose which restriction endonuclease you want to use to cut the vector and to put your DNA inside it. And then we talked about insertional inactivation. So be ready for a question on insertional inactivation in boards. If it comes, it could be a 3-marker question easily to explain what is insertional inactivation. So do you remember insertional inactivation? Iram, Parker? Anyone remembers what was insertional inactivation? Mhm. What does the word say? You are inserting something. What are you inserting? The gene of interest. And because of that insertion, which is happening, there is something getting inactivated, right? So the inactivation happens of this gene, beta galactosidase. So we insert our gene of interest anywhere between in this gene. So when how to know that our insertion has happened? If this gene is inactivated, then our insertion is there. So if the plasmid is transformed properly, our gene must have got uh, between this gene. So this gene is now inactive because it's cut into two and our gene of interest is in between. So you will see colorless colonies, which means beta galactosidase is inactive. And those will be the transformant colonies. And if you get blue colonies, which means beta galactosidase is active. So when it is active, it gives a blue color, which means those are non-transformants and our DNA recombination method has not worked. Okay, so this is a good alternative uh, in place of using antibiotic resistance as a marker. Remember antibiotic resistant genes used as markers? We talked about it. In biotechnology, whenever you make a plasmid, you put a marker gene. What is a marker gene? Yes, Parker, go ahead. What is a marker gene? As the name suggests, that will help you know it's a marker. So it will help you to know if your transformation process, the process of making recombinant DNA is successful or not. If it is successful, then your plasmid carries a antibiotic resistance gene. So if your plasmid has successfully entered inside the bacteria in which you want to, culture and make multiple copies of your gene in, then the bacteria, because of getting the plasmid and because of getting your gene of interest, also must have gotten antibiotic resistant gene. So if you now try to grow these bacteria in a antibiotic condition, they will grow. So if they grow and survive, they are transformants. If they don't grow, they have not received your plasmid. But then there are some issues with using antibiotic as a marker, antibiotic resistance gene as a marker, because unnecessary exposure of antibiotics to bacteria can make them resistant in nature, and more resistant bacterias cause problem, right? So disease causing bacteria, if they get resistant, then it's a problem because we cannot treat the bacterial infection effectively. Okay. So people thought that instead of using that, let's use insertional inactivation method, where other gene, we can insert our gene of interest into that. And if it gets inactivated, we'll get to know when we will plate the colonies. If the colony gave a blue color, it's non-transformants. If the colony gave a colorless color, which is like no color, or white. So people sometimes call it blue-white screening. If you get white colonies, your transformation has worked. If you get blue colonies, it has not worked. Okay, makes sense? Parker, thumbs up if you understand this. Okay. And then we talked about uh, making the host cell competent, because competency is the what you can say, it's the ability of the host cell to take up any foreign DNA. Otherwise, normally cells do not take up DNA when given from outside. Only 1%, 1 to 2% of bacteria in their natural environment can take up a foreign DNA. Rest 98% bacteria cannot. And so if we want to use bacteria to clone our gene, then we have to make them, in most cases, you have to make them competent. And to make competent, there are many methods: heat shock plus using divalent cation, which is the most commonly used one. Microinjections, gene gun are other methods that we talked about in detail. So just to give a summary, heat shock plus divalent cation is used mostly for bacteria. Okay, microbes. Microinjection, where you directly inject recombinant DNA into the nucleus of a cell, is used for animal cell, because it's easy to insert a microinjection in an animal cell, but not in a plant cell. So for plant cell, we use gene gun, or which is also known as biolistic method, right? Where nanoparticles coated with DNA are bombarded on the cells, like lots of bullet being fired, nanobullets. And then fourth is the disarmed pathogen vector, where we take a natural vector, a natural microbe, which has the ability to transfer a piece of DNA into a cell. And these are pathogens that are viruses and also bacteria sometimes. So we use that pathogen, and by disarming, we mean we remove their pathogenicity. So they are no longer lethal, but they can do the work still, which is transferring the genetic material to the host cell. Okay, so that's using a natural method after taming it. Okay, cool. And then we basically talked about the whole process. And yes, PCR was one such thing that we talked about. So, sorry, do you remember PCR, people? Parker, Iram, what is a PCR? And when do we use
Polymerase chain
it?
reaction.
Yes, right, Iram. And when do we use it? When do we need to use it?
when we want to get multiple copies of the gene of interest.
Yeah, when in that whole process. So you started with isolation. So DNA extraction you both did in the practical. That is the first step of doing biotechnology also. So you extracted DNA. Then you also used restriction endonuclease to cut down your gene of interest. You also used gel electrophoresis to separate your specific gene of interest. And you have your DNA in your hand. When will you use PCR if you want to use it? So you said it is to amplify our DNA, right? Amplify the copy numbers, correct?
Yes, sir.
So when do you think we will ideally need to do that? We will need that before we insert our gene of interest into the plasmid, right? Before inserting into the plasmid, we would like to make multiple copies of our. So we have, and mostly it is done in cases where DNA extraction and gene isolation has not worked. The yield is not very good. So like Parker used kiwi to extract the plant DNA. You used banana, right, to extract the plant DNA. Let's say you both had to extract a gene out of that DNA fragment. But to begin with, if your DNA extraction methods were not very great and clean, then the yield of DNA will be low, right? You will get less DNA molecules extracted. Correct? You understand? And just because you will get less DNA molecules extracted, you will also have less copies or less number of DNA fragments of your gene of interest. Agree, both of you?
Yes, sir.
But you need, let's say, millions of copies so that you can make millions of plasmid and you can increase the chances of your recombinant DNA recombinant technology being successful. So for that, even if your yield was low, no problem. You don't have to go back and do extraction from the beginning and then again use restriction enzyme to isolate, cut the fragment, then do gel electrophoresis to isolate. No need to do all that. If you have less number of purified DNS fragments, then you do PCR on those and you can increase their number, right? So we need it after we extract the fragments, we isolate, purify the fragments, and before we make uh, a recombinant DNA plasmid. Make sense, both of you? Yes? No? Okay, perfect. So just remember the basic steps of PCR. They often ask you, sometimes they give this diagram only in the exams and ask you to label the unlabeled things. So which step is what? So basically there are three steps: denaturing, annealing, and extension. So denaturing simply is opening up of the DNA, both the strands. Then annealing is you want your primer. So there are two primers, forward and reverse, to bind on those strands. And then you want extension. And once the extension has happened, then you, in one cycle, you get two copies. And then in two cycles, you'll get four and so on. Correct? So just remember their temperatures also, and the name. That's it. And to do the last thing, which is large scale synthesis, we call it biosynthesis stage. There are big bioreactors that are used. And we talked about what are bioreactors and how do they work. So basically, it's a big vessel where you keep the bacterial culture and keep giving it air and frothing so that it continues to grow more and more simply. Okay, that's called downstream processing. So this was the principles chapter. In applications, whatever you have studied in principles, we are just going to apply it in two fields, agriculture and medicine. And I think this is where Iram had a doubt, right? In agriculture, right? using what was
Yes.
what out?
RNA
RNA
interference.
interference, right? Okay, perfect. So I think we did RNA interference for um, yes, pest resistance tobacco plant, right? Yeah. So this is just an example, but you can apply RNA interference for many other different things also. So simply, what do you understand, Iram? Where is your understanding? You tell me what do you know about RNA interference, and then I'll pick from there. Whatever words is comes to your mind, what do you understand?
So there's like a nematode that is
Mhm.
known as Melarogini incognitia that infects the roots of tobacco plant, and
So that is the problem, right? So you have
yeah, this
identified
is
a
the problem.
problem that there is a nematode, which is a worm called Meloidigyne incognita. And Meloidigyne incognita, it affects tobacco plant, basically the roots of tobacco plant, and kills the cells of the root, right? So basically it makes the plant weaker and weaker, and over time the plant will die. So there will be less yield of tobacco. And tobacco being a commercially very successful plant, so there is a whole industry around it. So obviously people who work in tobacco industry do not want their tobacco crop to be destroyed. So this is the problem, right? Now they wanted to
Yes.
get rid of this problem. Yes. So this is where RNAi technology came in. Yeah. So what do you understand of that? What
So
was done?
like what we do is the mRNA, this disease or this nematode consists of this mRNA which is causing the problem, and we silence this by a complementary DS RNA.
Sorry, which mRNA did we target again?
So the mRNA which is present in the nematode, right? We
Right.
silence
So yeah, so there will be many mRNAs present in the nematode. There will be thousands of gene and there will be thousands of mRNAs.
the mRNA specifically causing the disease.
Not so the mRNA that's specifically causing the disease, um, it's produced in nematode, but it is secreted. Uh, uh, if there is a mRNA that is causing the disease, it it will come out in the nematode cell, will make a protein, and the protein will cause the disease, right? In the tobacco plant.
Yes. Yes, sir.
So we don't know what that gene is. It could be hundreds of different proteins that are causing problem. So will you target, let's say, even if there is 10, there won't be just one gene that causes disease, right? Nematode might be secreting 10 different kinds of things. One is a toxin, another is something else, another is something else. So how many will you target? And let's say we don't know which of the toxins produced by nematode is causing the disease in tobacco. Maybe one toxin is affecting the root, the other toxin can do something else. But we know that the reason that they are able to cause infection, what is the reason? Reason is that nematode, when it goes inside and affects the tobacco plant's root cells, it divides and multiplies there, right? A single nematode cannot do anything. Do you agree?
Yes, sir.
For any infection to occur, there is a concept called threshold load. It's true for COVID. It's true for any other bacterial, fungal, viral, protozoan infection. We all have been exposed to COVID virus. All, even if you think that you were closed in a cabinet all these 2.5 years, still some amount of COVID virus must have entered your body. But we didn't get COVID. Why? Because it was not enough to cause an infection, and our immune system could kill it before it could overwhelm our system, right? But when someone comes into contact with a, a, a person who's infected, the amount of virus that passes on is too much, and that can cause a problem. So it's all about load. Okay. Same is with bacteria. We all might have some bacteria sitting inside our lung, but in very few numbers, and they are unable to, you know, pull up a whole rebellion or a heist to hijack our system. So immune system is keeping it in check. Every day, millions and millions of dust particles, viral particles, bacterial particles enter our body, but we don't fall sick every day. Make sense? But for these infections to happen in a systematic manner, the worm has to go inside, sit inside the cells and the tissues, and replicate and divide. So instead of targeting the toxin that it is producing, what if I target something so crucial, a gene which is very, very important for nematodes' own growth and development and multiplication. So if I silence a gene in nematode without which nematode cannot survive, okay? And if nematodes don't survive, then infection will also not survive. Does it make sense, Iram?
Yes.
And that way I can just select one gene, one very vital, crucial gene. If you have to select one gene which you perturb in humans and humans won't survive, then you will probably look for a very crucial metabolic gene, right? Because metabolism is very, very important. You might just you know target the gene hemoglobin, correct?
Yes, sir.
If you
But
block
sir,
all the hemoglobin, yeah, go ahead.
there are so many genes. So how do you identify?
Correct. There are so many genes. That's the problem. So we just need. You mean how do I identify where is the gene for hemoglobin?
Yes.
Yeah, yeah. So for that we can only do such things in organisms whose genome has been sequenced. So for nematodes, worms, these are small organisms. So you just take the sample of this causative agent. Then you first have to sequence the genome, and then you get to know that it has so many numbers of chromosomes and so many number of genes, and these genes are present on this and that. Then you can target one particular gene. Because to target a gene, you need to know the sequence of the gene, right? Without knowing the sequence, you cannot target the gene. And to know the sequence, you have to sequence the genome. So human genome sequence, remember Human Genome Project was to sequence human genomes. We have sequenced lots of animal genomes, especially genomes of um, bacteria, pathogens that cause disease, and also for animals that are model systems for research, and also for useful organisms, both plant, animal, and microbes. So thousands and thousands of organisms have been genome sequenced. So because of that, we know the sequence of a very crucial gene. So what we will do is um, there will be two DNA strands. Okay, we will, and first, where is all this happening? Where is this fight happening? It's happening inside a plant cell, correct?
Yes, sir.
And what are the methods of introducing DNA into a plant cell? We can either use a natural vector, which we have, um, you know, changed or modified for our benefit. So we know already that there is a pathogen that is very, very successful in sending or infecting the root cells of plants. That's called Agrobacterium tumefaciens. So we use that pathogen as a vector here. So we disarm the pathogen, we make it not lethal, so it will not cause infection or disease. But what we will do is we will take the plasmid out, and instead of its TI plasmid, which is called tumor inducing plasmid, in that plasmid, we will take out all the lethal genes, disease causing genes, and we will insert the sequence of DNA against the gene of the nematode. Okay, two strands, sense and antisense. So basically why we need to send two strands, so that when this DNA goes inside the plant cell, it will produce two RNAs, a sense RNA, which will go in 5' to 3' direction, let's say, and the antisense RNA, which will go into 3' to 5' direction. And that will join together to make a double-stranded RNA, dsRNA. Now, this dsRNA that we have made, it will be specific to a nematode gene. Can you see on the board? And this gene will be the gene that we have selected to target. Now, this DS, whenever there is a double-stranded RNA in a living system in eukaryotic cells, it is not a good sign. So double-stranded RNA is a signal of an issue. And this employs a risk complex, RISC. Okay. And this risk complex will go and find that mRNA against which this dsRNA has been developed. And that mRNA will be then tagged, identified by this risk complex. It's a complex of proteins. You don't have to go into the detail of it, but this is how RNA interference works. So RNAi simply means using RNA, you are interfering another RNA. And that RNA was an important RNA for a gene, crucial for the survival of nematode. And that mRNA will be tagged and destroyed. So, and let's say that mRNA was a very important mRNA for metabolism of the nematode. Now there will be no metabolism happening and nematode will die. And this will happen everywhere. All the nematodes will die. So when they will die, their numbers will reduce and the infection will be cured. Is it clear, Iram? So to cut it short, we send two strands of DNA, sense and antisense, DNA1 and DNA2. They both are loaded in the plasmid of Agrobacterium tumefaciens. And it can easily send this DNA inside the plant cell. In this case, tobacco plants root cells. When they will go inside, the DNA will produce RNA. So there will be two RNA, sense RNA, antisense RNA. They will join together to form a dsRNA, double-stranded RNA. That double-stranded RNA will be like a signal in the cell for a problem. And that double-stranded RNA will be specific and complementary to a gene in the nematode. So double-stranded RNA will deploy a risk complex. They will identify that mRNA of the gene, tag it, destroy it. And that way, you are blocking the expression of a very important gene, important for, crucial for survival. So nematodes cannot survive. Is it clear, Hiram? All? Any doubts?
Yes, sir.
Okay, perfect. So let's quickly go and look at some questions, okay? Since today's class is going to be a shorter one, so we have around 35 minutes to spend on questions. So I think after a revision, let's go. I'll stop sharing this. And I will share this screen. Can you see this screen that I have shared? Yeah.
Yes, sir, I can see.
Okay, perfect. So yeah. So I've picked some questions. So you just read the question, no need to read the options, and then take a minute and tell me the answer, okay?
Okay.
So these are easy ones in the beginning. For example, the term molecular scissors refers to, it's for restriction enzymes. And sometimes in the options, there will be uh, endonuclease instead of restriction enzyme. So you have to choose that. It's also called chemical knife, molecular scissor. All these terms are used for endonucleases. Okay.
Yes.
And things. Yes, question 19. So this is from NCRT only, and this has been asked. Can you see question 19 clearly?
Yes, sir.
Just read it for yourself and tell me the answer. Try to be correct and not very fast in the beginning, but yeah.
Study the following figures and identify the enzyme involved in step 1 and step 2. Number 1, eco R1 and
Don't
DNA
read the
ligase.
options. Just look at the figure and tell me which option you think is correct.
Option A.
Option A, right. And why not option 2, B? So you are right if you thought that, see, first what's happening is cutting, right?
Mhm.
So you need
Yes.
a endonuclease.
Restriction
And
endonuclease.
second is pasting, so you need a ligase.
DNA ligase. Yes.
Yeah. So A and B both, the first part is our endonuclease and the second part is DNA ligase. But why one and why not two?
Because eco R1 will recognize the sequence AATTGC.
Yes. Yeah. So GAATTC, what you are seeing here, GAATTC is recognized by EcoR1, and EcoR1 cuts exactly between G and A. Okay.
Yes.
Yeah. So just remember that. Cool. So this is exactly this is from NCRT only. Yeah. Question 27. It's just simple but trick question. Will you read the question? Are you there, Iram? Question 27.
Yes, sir. How many fragments will be generated if you digest a linear DNA molecule with a restriction enzyme having 4 recognition sites on the DNA? Mhm. D.
D. 4.
Yeah, not
Is it?
sure.
Yeah. Okay, what's there to be not sure? It's very simple question. So, and these are fairly common ones. So these kind of questions often come, what they will do is just change number from 4 to 3 or to 5 or something. But the concept lies in 2 words. You have to carefully understand whether they are talking about linear DNA or they are talking about a circular plasmid DNA. That's the only catch in the question. So in this case, what are they asking? They are digesting a linear
Linear.
DNA molecule. Now remember one thing. if a DNA is circular, okay,
Hmm.
and then you cut it once, so it's like a ring. If you
Yes.
make one cut in a ring, how many fragments are produced?
One.
One, right? Because the first cut in a circular DNA is just to linearize it. You opened the ring, but ring was also a single fragment. And when you cut it, it is still a single fragment, right?
Yes.
So the first cutting goes like waste in a plasmid. It does not increases the number of fragments. But if you are starting with a linear DNA, it is already linear. First cut will produce two fragments, correct?
Yes, sir.
Yeah. So if it is a linear DNA they are starting with, then the number of fragments will always be one more than the number of recognition sites. So if you are starting with linear DNA and you have four recognition sites, which means four places where the restriction enzyme can cut, then the total fragments will be five. So in this case, the right option is C, 5, because
Does
it's a
that
linear DNA.
happen? It doesn't make sense.
Yeah, it can happen. That's where, so first it's a hypothetical question, but it can happen if you take a long piece of DNA. So in our DNA, in my DNA, if and that's how we can actually make fragments of DNA. So there will be millions of places where EcoR1 can recognize GAATTC. There will be multiple places in my genome where GAATTC will be present, right? You understand?
Yes.
Yeah. So if you take a linear DNA and you have four recognition sites, then there'll be five total fragments. But if instead of the word linear, they use a circular DNA here or a plasmid DNA. So if the word is circular or plasmid, then the number of recognition sites will be equal to the number of fragments. So if it was a plasmid DNA or a circular DNA, then the answer would have been D, 4, because there were four recognition sites. Make sense?
Yes, sir.
Yeah. So very simple, but often, either you lose marks or yeah. Yeah. Questions like this comes from gel electrophoresis and other techniques like PCR. So would you like to read 33?
Yes, sir. Having become an expert on gel electrophoresis, you are asked to examine a gel for a colleague. Where would you find the smallest segment of DNA? Near the positive electrode, farthest away from the wells, near the negative electrode, close to the wells, near the negative electrode, farthest away from the well, near the middle. They tend to slow down after first few minutes. Near the A,
Okay.
option
A.
B,
And why do you think it's A?
because it moves to the positive terminal, right?
Right.
The farthest one. So A.
Yes. So whenever we run a gel, the negative terminal has to be where we load the sample, because we know that DNA is also negative. So we will keep it at negative terminal so that it can move towards positive, and in that process, it can resolve into different fragments. And the smallest one will move farthest away from the well and near to the positive electrode. Yeah,
Yep.
perfect. And 34. So this is not directly talked about in NCRT, but a question that is from gel electrophoresis.
Okay.
Yeah.
Which of the following steps should be performed by a person in order to visualize the bands of DNA fragments obtained from gel electrophoresis? Exposure of DNA fragments to UV light, staining with bromophenol blue, followed by exposure to UV light, staining with ethidium bromide, followed by exposure to UV light. Person can see the bands without staining. Option C.
Yeah. So
It's given in
sim
NCRT.
oh, it is. Okay, cool.
Yeah.
So see, to visualize DNA, you need two things. You need something to bind to DNA that can be converted into a chromogenic effect, right? So DNA itself does not illuminate. DNA itself does not give out any color. Okay. If you just use UV on DNA, it will break simply. Nothing will happen. So you need to first stain it with something. And ethidium bromide is something that goes and bind to the grooves of DNA. Okay, minor groove. So groove simply means, remember I told you DNA is coiled and all. So there are some regions where this ethidium bromide goes and binds. Okay. And then when you give UV exposure, that UV is absorbed by ethidium bromide and it gives a color. Okay. So it illuminates in the presence of UV. So basically what you see is ethidium bromide. You still can't see DNA, but you know that ethidium bromide is bound to DNA. So wherever will be ethidium bromide, there will be DNA. Make sense?
Yes, sir.
Yeah. Okay. Um, let's do this one last question about gel electrophoresis, and I think then you are good to go. Question 35. Yeah. Yeah. I think you can see it, right? Yeah.
Yes, sir. Study the given figure carefully and select the incorrect statement regarding this. It
Yes.
represents a
Don't
typical.
read the options. Just look at it carefully and tell me the options. Read it for yourself. Just read the question and then read the options for yourself.
So option B.
Option B. Yes, correct. So I think you are now ready to tackle
Yeah.
electrophoresis related questions. So we'll try to go to some other concepts. Um, yes, question 38. See, so this chapter has a lot of concepts, right? So it's very easy to make questions from this chapter. Pick up any concept and write some statements and ask whether it's true or false, right? So expect these kind of questions coming from this chapter, biotechnology, and also from evolution. There also you are introduced to a lot of concepts about evolution, um, theory of natural selection, Lamarckism, Darwinian, etc., etc. So they will just write some fragments and then, sorry, they will just write some sentences and then they will say whether it is correct or incorrect. So question 38 is one such question, and it says which of the following statement are correct. So just read it for yourself and let me know what do you think is the right answer.
So I think everything is wrong. Option
What?
B.
Option B. Yes, you're right. All the statements are wrong. Okay. So And I think if you understand everything is wrong, then I also guess that you understand what has to be the correct. Are you confused in any of the statements? Do you know the correct version of every statement? Because that's what is important to learn. You're right that all are incorrect. So you know that what is the incorrectness, like how all can be correct.
So this statement says that which one is correct, right?
Right. So you said all are correct, right?
Yeah,
Yeah.
except the last one, fifth.
Oh,
I did not see the fifth one. I just saw till fourth.
okay. Yeah, the fifth is very close to this thing. Okay, right.
Yes,
So you are saying one, two, three, four are correct, and fifth one is
correct.
incorrect. Right.
Yes.
So which option? So option starts A and B, C, D. So I guess you said option B
Yeah.
is the correct option you will
Yes.
choose
Yes.
because
But
one,
I
two, three,
chose
and
B
four are
because
correct. Yeah. You chose
yes.
B because
I chose B because in the B option we don't have like the fifth option. Though I'm not still convinced with the second option, hint to always cuts DNA molecule. I'm not so sure about it. But fifth is not a correct one, right? So B does not consist of fifth one. So I chose that.
right. And that's a very good technique. See, often these kind of questions are lengthy to attempt. So what students do is they just leave thinking that, oh, I'm not, it's five options and then I have to think about everything. But that's not true all the time. Even if you know, just like you figured out that, see, option fifth is for sure not correct. So any option that includes fifth is not going to be the correct option to choose. A, C, and D all have fifth, right? And fifth is an incorrect statement. So even if you are not sure about first, second, third, you can still choose option B because fifth is not there, and fifth is something you know is incorrect. So in a way, that's where I say that exams or entrances are also about keeping your um, composure. Like you have to stay calm and you have to manage time well. That's it. Even if you know everything, you can mess up exam if you do not manage time properly or if you do not keep your calm. Okay. And even if you don't know everything, in this case, like you said, you are not sure about uh, second option, that hint to always cuts molecule at particular point, and it recognizes a specific sequence of 6 base pairs, but still you know that fifth is incorrect for sure, so you could still choose the right option. Do you understand? So it's worth giving um, questions like this a try. Okay. And there will be a significant amount of question like this. Okay, let's move towards something more complicated, which is, so these kind of questions don't come nowadays in NEET. They were trademark of no, these kind of questions still come, but because they are not assertion reason, but just which statement is correct and which is not. So would you just read question 45 for yourself and tell me if you understand which is the right option?
Sir, option A.
Yes. A, both the statements are correct, right? Yeah,
Yes.
you're right. So, and in AIMS, when AIMS used to conduct its own entrance, there was a section of questions like this, a little more difficult than this, which was called assertion reason. So there'll be two statements there, and one will be
We have
assertion,
assertion
a
reason
sentence.
in both.
You have, yes, exactly. So where they give you one sentence, call it assertion. They give you
Yeah.
another sentence, and you have to then
Relate
figure
both of them.
out. Yeah, you have to figure out first if both are correct. Second, if both are correct and the reason is the correct reason for assertion. Third, if both are correct, but the reason is not the correct reason for the assertion. And then fourth, one or the other is not correct, incorrect, right? But
Yes.
this is more straightforward. This is like whether the statements are correct or not. Yeah.
This is not so bad as I thought.
Yeah, it's not that bad. So see, thing is a lot of a lot of times we get intimidated by entrances. We get intimidated by the fact that, oh, I'm sitting in an entrance and there are thousands and lakhs of students I'm competing with. And that kind of gets onto our nerves. And I've seen people very well prepared, being uh, very regular throughout the year, good with learning, memory power, and still messing up entrances. But if you plan it, and if you just know that, okay, this is how I'm going to proceed in the entrance, and I have a plan for it, you can do fairly well, right? And entrance is not about either you have to top it, like, there are 100 questions, I have to do all 100 correct, or else I have no chance. It's not like that. You just have to pre-plan that, see, this is what I'm going to attempt minimum. I need minimum this to achieve what I need. Okay. Um, so that should be your plan in the mind. What do you need to achieve what you want? So I want let's say I want a seat in this college or something, and this is what is needed. And this is how I have planned all my entrances that I have given for getting into institutions, right? So for my masters, when I gave the entrance for a very prestigious institution in India, it's called Tata Institute of Fundamental Research. I'm not sure if you have heard of it or not.
Sir,
So
it's in
it
Mumbai.
it's in Mumbai. Yes, you
Yeah,
have
I
heard
know.
of it. Yeah,
Yes.
so it's for a research master. So those who wish to have a career in research, it's a very good place to go and learn um, biological research and all sciences from. So it used to have an entrance and a pretty straightforward one. They will give you 60 questions and they'll give you 3 hours. So you might think, okay, just 60 question and 3 hours. Very, very opposite to what entrances like NET and NEET are like, where there are 180 questions sometimes and 180 minutes. So 1 minute per question. So planning becomes very different in these two kind of entrances. But then I knew that I don't have to do all 60, but I need to be sure that I should get 65, 70% correct marks. That's all. So, I just planned like I'll do 42 questions. I did 42 questions and I didn't even try or attempt others. Like I read them and I realized that there are chances if I attempt this, I might end up choosing the wrong answer because I do not exactly know how to find the right answer in that moment. Or if it is a factual question, I do not know which is the right option. So I thought not to select it. Just did 42, 41 were correct. And that's where you get what you want, right? So it's a lot about planning. That's what I'm trying to tell you. You understand? So do not get
Yes.
intimidated by the fact that you are giving an entrance, but plan it. Plan it well in advance. And don't think how many people, how many seats, and all. Just do the best you can, and that's all. Okay. And again, remember, see, no entrance is end of the world. Um, and I'm just saying it because if you don't get somewhere, you will get somewhere else, or you can get there by preparing some more. Okay. So don't let it stuff in your head and pull you down. So it shouldn't be like a weight. Most of the times, studies, entrances, giving exams pull very bright students down. So it acts like a weight on their legs that pulls them down in the water. That shouldn't be the case, right? you are a bright student, you like studying science, you understand logic, you like studying logic, you will end up getting somewhere or the other, right? And I think in the beginning we were also discussing sometime career options in science. So gone are the times when there were just two roads to travel. You take non-med, go and become an engineer. You take biology, go and become a doctor. So gone are those days. And these days there are entrances for a lot of different careers, career in research, career in biotech, career in pharmaceuticals, career in, not just being a human doctor, but being veterinary doctors, career in counseling, scientific advisors to companies, career in experts, biological experts. So there are companies like that make cosmetic products, and they always need biology experts who have studied biology, um, some discipline of it in detail in the masters or in PhD. So you can choose a lot of you can choose from a lot of options is what I'm saying. Okay. But
Yes.
again, like in all the options, you will have to go through one or the other kind of exam. So that's like a gate path. But the fact that you have more than one different kind of exam is good. No? Maybe maybe you're not the kind of person who can remember a lot of facts. So factual entrances is not your cup of tea. But when it comes to entrances that test your learning, test your problem solving skills, you can do better there, right?
Yes.
So that's where you have to plan entrances. So don't get overwhelmed at any moment. Okay. Okay. Cool. So there is something that some of the questions I have chosen, which are a little more complicated. Let's do a few of them. And since we have less time, so next class will be completely problem solving, both including both the chapters. Today I'm just doing principles. And one question is, mm, yeah, um, yes, question 100. So these long type of questions I have seen um, coming up frequently. Again, this is to intimidate you. Like you will see, oh, this question looks so long. Question 100. Can you see the question in complete? You can see that, right?
Yes, I can.
Yeah, just read and see if you understand this and what is the right option.
What is this? So A.
Yes, right, correct. And again, I think you understood the
So
trick to
with
do
just
long
two
questions
options.
is yes, right. So uh, you just have to find solution for one and then see that how many options have that exact same solution, right? So for example, even if you know this one, dash is a natural polymer extracted from dash, you know that it's agarose from seaweed. So just looking at this, option D is out of question now, right? Because it says size and sieving effect. So that's gone. Then you go to the next and you will see that one more option is gone. So that way you can do it faster, correct? Were you doing the same thing?
Yes, sir. Like the same.
Yeah, Cool. And uh, I just want you to solve one high order thinking skill. So these are questions that can also come in boards, and they're called HOTS question, high order thinking skill. They just require some more thinking and taking time. So question 3, can you just analyze this given diagram and tell me which colony has undergone transformation? We'll do this as the last question, or if you solve it fast, I will take one more HOTS and rest both the chapters. More questions we'll do next class, Wednesday. So this is a little time taking.
So 4C, option C.
Option C, colony 4, right?
Yes.
Yes, perfect. And would like to tell why
Yeah,
colony
because it
4?
was not
Because
able to survive in the medium. It just
ex-
vanished in plate N.
exactly. So, which means that it has undergone transformation because when the gene got inserted, the resistance for ampicillin got lost, right?
Yes.
Sorry, the resistance for tetracycline got lost because this is where the gene got inserted, right? You see this gene for tetracycline resistance, but when you inserted your gene to make recombinant DNA, this gene got inactivated. Again, insertional inactivation of tetracycline resistance. And when they plated it with tetracycline and ampicillin, this colony died. But remember, there is a catch here. What they can also do is, see, do you understand that this plasmid still has ampicillin resistance? Because the
Yes,
DNA
sir.
was inserted here. So if
Hmm.
they show you two pictures where agar with tetracycline is separate and agar with ampicillin is separate, you will see that colony 4 will grow in ampicillin but will die in tetracycline, right?
Yes.
So here it's a mixture of both, but just remember, you know that it's not ampicillin that's killing it. It is
It's tetracycline.
tetracycline that's killing it. Yes, perfect. So